So what I'm presenting today, like I said earlier, is a portion of my PhD titled Assessing Pharmaceuticals and Personal Care Products in the Greater Houston area. And then we assess the impact of the detected PPCPs on rat pituitary cells. So my presentation will take this outline from the introduction all the way down to the conclusions and recommendations. So I believe we all know what PPCPs are. So I'm not going to waste so much time talking about them. I'm just going to talk about the fate of human use PPCPs. As we all know, if we use pharmaceuticals and personal care products, we either excrete them into the toilet, flush, or we wash them off our body, and then they end up in the wastewater. And then when used in veterinary medicine, it also ends up washed off the animals and then ends up in nearby surface waters. And we know, well, PPCPs are biologically active agents because they are designed to interact with specific pathways within our body. And so far, most of them are considered to be endocrine-disrupting chemicals. So this shows the sources of PPCPs in our environment, ranging from manufacturing processes that can result in leaks or direct discharge, hospital and domestic sources from excretion, improper disposal, landfill, and then from sewage and wastewater treatment plants, livestock, poultry, and aquaculture, as I mentioned during the introduction. So my study area is the Greater Houston, and it is one of the largest metropolis area in the US with populations in the millions. And this area is known to host oil, gas manufacturing, and different petrochemical industries. As we know, these industries are contributing to the growth of this area, but they also result in polluting the area. So the region watersheds offer a series of services ranging from tourist attraction, flood control, and so on. And so we believe pollution of this area can lead to threats, can result in potential threat on human and aquatic organisms that we have in the watersheds. So this graph shows a summary of my watershed. I have 9 different watersheds that I looked at, and the heavily populated area, and the watersheds are located on the western boundary of the Galveston Bay, which is the most industrialized and heavily populated area within the region. So why do we decide to go for this study? We know as we use these PPCPs, we use them every now and then. At the end of the day, they end up in the environment on a daily basis. So because of this, they're kind of persistent in the environment. And so far, no investigation in this area has checked the presence of these PPCPs in water, sediment, fish. If they have, not at the large scale at which we are looking at it, because like I said earlier, we're looking at 9 different watersheds. And then I mentioned as well, most PPCPs are suspected endocrine disrupting chemicals. However, not a lot of studies really demonstrated their endocrine disrupting effect. So we seek to kind of look into these areas. So our aims, the first aims and objectives, the first one is to screen water, sediments, and fish samples collected from from the watersheds in the Greater Houston area for a contaminant of interest. In this case, I looked at 5 different PPCPs, and then we assessed the potential ecological risk of these PPCPs in the watersheds, and we also investigated the effects of the PPCPs we detected on physiological parameters in rat pituitary cells. So to be specific, we looked at hormone quantification in the rat pituitary cells, and we also looked at cell proliferative and cytotoxic effects of the PPCPs. So risk, the first, okay, I'm sorry, I think my slides rearranged so. So the methods we use for the analysis of the PPCPs, we collected the water and sediment samples around between 2020 and spring 2021. And the method for collection is stated here. For the extraction of the PPCPs from our samples, we use a solid phase extraction, slightly modified EPA method, and so I use this for my extraction because I mean that's what I could easily access at the point. And then we use the SCMS for the detection of the PPCPs in our samples. So for the ecological risk assessment calculations, I used this method by Zhao et al. 2016, calculated the risk quotient. So this slide here shows the reason why we chose to do the risk assessment. Like I mentioned earlier, the watersheds, they are very important to the ecosystem in this area. So any form of disturbance in this ecosystem can result in destruction of different aquatic organisms within the water, as well as the bleaching of coral that is kind of ongoing as a result of pollution. So we decided to go for the risk assessment part of the study. And then for the cell study, we use the RC4BC cell line, which is a adenoma cell line. And then we use the ABC TC4195 cell line, which is just a pituitary cell that is not diseased. And why did we choose pituitary cell? We know the pituitary cells are master endocrine, the pituitary gland is the master endocrine gland. It controls the activities of other endocrine glands in the body. We didn't have access to human pituitary cells, so we decided to go for what we have available. And this summarizes the cells of the pituitary cells, the hormones they produce, and the effects. I mean, the hormones as the releasing hormone, the hormones are secreted, and then the target organ. And as you can see from the slide, the hormones of pituitary cell affect pretty much everything within our body, from growth to the adrenal gland, thyroid gland, the gonads, and so on. And then for the cell proliferative and cytotoxicity assay, we use MCT. So this shows the potential pathway of endocrine disruption in the cells. So we have the genomic pathway. Since I'm not focusing on steroid hormone, my research was tailored towards the non-genomic pathway. So activation of the G protein coupled receptor can lead to a cascade of events that can result in inflammation or genetic modification down the line. So we also did joint action toxicity tests. This involved determining the concentration at which 50% of treated cells inhibited an effect, which is EC50 or IC50 as the case may be. And then this calculating the synergistic ratio model was supposed to be used to check for the joint action of the different PPCPs that we have. And then here are the hormones that we studied in our research. We looked at the rat follicle stimulating hormone, adrenocorticotropic hormone, thyroid hormone, and then to check for the activity of the GPCR in the cells. Like I showed in the pathway, GPCR is the membrane protein that is activated by potential hormone or endocrine disrupting chemical. So here we looked at the effects of PPCPs on CAMP, which is cyclic adenosine monophosphate. So to validate our method for extraction analysis of the PPCPs, we use the same method we use for the samples to analyze some spike samples, and then we calculated our recovery, and this table shows the recovery for each of the PPCP that we looked at. So the concentrations of our PPCPs, they were found as pretty large nanogram per liter level, and the highest concentration, as you can see from here, were found in, for most of the PPCPs, were found in Braze Bayou, and closely followed by the Hunting Bayou, which is the this bayou right here, and then we have in Greens Bayou. So here I took the liberty of showing the maps of the three bayous. So Braze Bayou showed the highest concentration, and we can see upstream of our downstream sample point. The green circles represent the sample points. Upstream of our downstream sample points, we have a lot of hospitals in this area. And the bayou on my right here is the Greens Bayou, and we can see a lot of wastewater outlets, which are the red triangles, as well as hospitals in the area, which could have impacted the concentration of the PPCPs that were found in the watershed. Then the Hunting Bayou watershed, not so much is going on. It's a pretty small bayou, but we believe as a result, because of the population of people that we have in there, the population to the bayou landmass ratio could have contributed to the high concentration of PPCPs that were found in the bayou. And then in our sediment samples, Sims Bayou showed the highest concentration of the PPCPs. And this on the right, I have the map of Sims Bayou, and not so much is going on as well in terms of hospitals, wastewater outlets, but we have some landfill location, and we have an airport there, which could have not resulted in the high concentration of PPCPs that we saw, but we believe because not so much is going on in the bayou, the PPCPs that are going in there kind of remain in there. That was assumption or conclusion from what we did. And then comparing the concentration of PPCPs in the water and the sediment samples, we can say that higher concentration of the PPCPs were found in the water samples compared to the sediments. And then in the fish samples, we found, we looked at Atlantic mackerel and we looked at the hardhead catfish. We found higher concentration of the PPCPs in the Atlantic mackerel. But when we looked at the we looked at the fillet as well as the skin. Fillet is the edible part of the fish. We saw that even though Atlantic mackerel showed higher concentration of the PPCPs in the skin as opposed to the fillet, while in the hardhead catfish, we saw more of the PPCPs in the fillet compared to the skin. So this is the take home from the first part, which is the analysis of the PPCPs in the sample. Higher PPCPs levels were found near the watershed outlets and hospitals around our sample point. PPCPs follow similar trend from upstream to downstream of each bayou. So say we found higher concentration in upstream, it is kind of consistent for all of the PPCPs that we looked at in each of the bayou. Then highest PPCPs concentration were found in Sims bayou sediment samples, as I mentioned earlier. And then significantly higher concentration of PPCPs were found in water compared to sediment and the fish samples. And of the two fish samples we saw, Atlantic mackerel accumulated more of the PPCPs compared to the hardhead catfish. And then for the risk assessment, I use those parameters, and this shows the result of my risk assessment. So CIP is CIPRO, SMX sulfametaxazole, NA-SMX is N for acetyl sulfametaxazole, which is a metabolite of sulfametaxazole, losartan potassium, and metoprolol titrate, and chlorfibric acid. So as we can see from here, we have low risk to moderate risk, even though LSP, SMX, they show pretty high concentration in our sample. Here, they're not posing as this risk posed by this PPCP is not as high as we have for metoprolol and then titrate and then the N for acetyl sulfametaxazole.
1.5 minutes remaining.
I have 2 minutes?
Yes. Okay,
so I'm just going to summarize the result of our cell assay. We did proliferative and cytotoxicity assay. For the proliferative assay using the first cell line, which is the adenoma, we saw the metoprolol titrate increased the cell proliferation after 24 hours, and we didn't really observe so much change at 48 and 72 hours. CIP promoted cell proliferation at all time points. And then for cytotoxicity assay, we saw various effects in the RC4B cell line, no consistency were observed. And then for the second cell line, which is the one that is not diseased, we saw cell inhibition of cell proliferation, and then we saw increased cell proliferation for metoprolol titrate, while the other inhibited cell proliferation. For the cytotoxicity assay, we saw time-dependent cytotoxicity, which is death of cells after 72 hours. As the time increased, the cell deaths were increased, but not so significant. So based on what we did, we didn't really see any consistency in the first cell line, which is the adenoma. So we decided to go for the cell line that is not diseased. And this shows the proliferative assay and cytotoxicity assay that we did. Of course, nothing significant really happened, but we saw variations in proliferation and cytotoxicity. And then for the hormone levels, various effects were observed. I'm sorry, I have to rush through this. For FSH, no significant effects was observed at all. And for ACTH, in some cases, we have inhibition and then we have stimulation. Same goes for the CAMP. So based on this, we concluded that we saw time-dependent effect of PPCPs on cell proliferation and cytotoxicity, which kind of shows that the endocrine disrupting effects of this agent or these chemical compounds can be way complex than we thought. And then dysregulation were observed in production of different hormones. And then recommendation, environmental regulation and monitoring. We know not so much attention is being paid to PPCPs because they believe they are found at very low concentration. Maybe proper industrial and waste disposal practices, proper waste treatment, fish consumption guidelines, and then, of course, more endocrine disruption research needs to be done. I would like to thank my mentor, my colleagues in the lab, College of Pharmacy, and then the funding source, Title III Fellowship, University of Houston, and then the Texas Southern University Office of Research and Innovation funded the research. Thank you. Any questions?
I will encourage everyone to ask questions on Hupe as we are out of time.